Good morning, everybody. My name is Brigette Romero. I'm a third-year PhD student in the Medical Molecular Sciences department, and today I'm excited to share a glimpse of my research with you. Did you know that cerebral palsy is the most common motor disability in childhood? About 1 in 345 children in the US have been diagnosed with CP. This condition is caused by abnormal brain development or damage during early stages of growth. And while nervous system lesion is non-progressive, the neuromuscular function often deteriorates over time, leading to contractures, spasticity, and weakness. Reason why the children with CP often require ongoing physical therapy and even surgery to reduce the pain that is caused by these contractures. Currently, there are 4 different types of CP. The most common type, and the one that I'm currently focusing on, is a spastic CP, which accounts for more than 80% of all CP diagnoses. As you can see in this figure below, there are some differences in the skeletal muscle in patients with spastic CP and typical developing children. Kids with CP have a smaller muscle fibers, fewer satellite cells, and an increased extracellular matrix. It's also reported that the sarcomeres are not added in series, and as a result of that, kids with CP have this overstretched muscle, which difficult the mobility as well. However, the molecular and cellular mechanisms contributing to this motor dysfunction in spastic CP are still largely unknown. And despite all these small findings, currently there is challenging to diagnose CP. This diagnosis is often delayed for several months or even years until these symptoms become more apparent. Diagnosis mostly rely on clinical observations and imaging techniques. However, approximately 40% of the children diagnosed through these techniques do not go on to develop CP. That's why there is a pressing need for a specific method that can identify the disease at that early stage. And during this journey of finding a new diagnostic method, we came across regulatory RNAs. These regulatory RNAs have been shown to play important roles in various other diseases, including musculoskeletal disorders, and they are potential candidates with roles in CP that are just beginning to be appreciated. These regulatory RNAs are approximately 98% of the RNA family, and they come in different shapes. But for today's presentation, I just want you to focus on circular RNAs and microRNAs. Circular RNAs are the most recently recognized and least studied member of the regulatory RNA family. They are known to interact with other regulatory RNAs. Particularly, they expunge microRNAs and prevent them from binding to other targets within the cell. Because of the circular structure, they are pretty stable, around 5 times more stable than mRNA, and they have become great candidates for non-invasive diagnosis. In the coming slides, I will be explaining how we could use circular RNAs as diagnostic biomarkers. For us, the first step was to validate circular RNAs in cells. For that, we obtained muscle samples from CP and typical developing children. We isolate satellite cell-derived myoblasts, we grow them in the lab, and we evaluate the expression of different circular RNAs. After confirming our findings by PCR, we found that this particular circular RNA, known as circular RNA nuclear factor 9, was shown to be present at lower amounts in the cells from CP patients. This circular RNA is highly expressed and participates in the development of many organs of the human body, such as brain, heart, and muscle, and is currently being used to diagnose cardiovascular diseases and glioma. But since the function of circular RNAs depends on their localization, we need to understand and know where they are located. And for that, we use searchfish technique to visualize at high resolution the presence of this molecule. And we confirm that the circular RNA is indeed downregulated in CP compared to typical developing. And we found as well that this circular RNA is mostly cytoplasmic and could interact with some microRNAs. After bioinformatic and in vitro studies, we found that this circular RNA interact with microRNA 373. In the context of cerebral palsy, since there are lower amounts of this circular RNA, more of this macron is going to be available to go and bind to the MEF2C UTR. And as a result of that, the translation of this MEF2C protein is inhibited. And this protein is important because it's a transcription factor that regulates the expression of different sarcomeric genes, and thus could explain why kids with CP have this overstretched muscle. But as I mentioned at the beginning, the overall goal is to use it as a diagnostic biomarker. And for that, we plan to increase our sample size, and we aim to collect blood samples from different CP subjects and typical developing children, including individuals of different ages, genders, and disease severity at the time of diagnosis. In that way, will allow us to, just by taking a sample of blood, identify the condition at early and specific way. In summary, this approach will not only allow us to diagnose the disease early on, but also allow us to do an early intervention, which allow for better long-term outcomes and to improve the quality of life of the patients. And of course, will let us to develop customized treatment plans. Finally, I just want to thank members of Patish and Akin's lab, as well as the Cerebral Palsy Center and patients, families, and our founders. Thank you so much. I will answer any question.